Let's continue on our discussion on anemia of chronic disease. When anemia of chronic disease develops alongside chronic inflammatory conditions, with rheumatoid arthritis being the most frequently cited example, the laboratory picture can become significantly more complex and potentially misleading. In these inflammatory states, the anemia may present with low serum iron, reduced red cell color known as hypochromia, and smaller than normal red cells known as microcytosis. This combination precisely mirrors what one would expect to find in iron deficiency anemia. The resemblance can be close enough to cause genuine diagnostic error, and misidentifying anemia of chronic disease as iron deficiency anemia carries real clinical consequences, particularly if unnecessary or ineffective iron therapy is initiated. The critical distinguishing feature lies in the total iron binding capacity, universally abbreviated as TIBC. TIBC reflects how much iron the blood's carrier proteins, primarily transferrin, could theoretically transport if fully loaded. When the body is truly iron deficient, it responds by producing more transferrin, essentially dispatching more carrier proteins to scavenge whatever iron can be found. This causes TIBC to rise. In anemia of chronic disease, however, transferrin production is suppressed as part of the broader inflammatory response, and TIBC is characteristically decreased or low. This single laboratory finding, TIBC moving in opposite directions depending on the underlying diagnosis, is one of the most reliable and clinically actionable ways to separate these two conditions from each other, and it is considered a cornerstone distinction in hematology. In summary, anemia of chronic disease, also known as anemia of inflammation, is commonly seen in chronic inflammatory states, including infections, autoimmune diseases, malignancies, chronic kidney disease, heart failure, and obesity, and is primarily mediated by inflammatory cytokines and the iron-regulating hormone hepcidin, which together impair iron availability, suppress erythropoietin production, and inhibit erythropoiesis. Key features include normocytic or mildly microcytic anemia, low serum iron, low or normal transferrin, elevated ferritin, and iron sequestration within macrophages, with diagnosis largely by exclusion of other causes and characteristic laboratory findings. Laboratory diagnosis. Hallmarks are low serum iron, low to normal total iron binding capacity, TIBC, elevated ferritin, normal or low reticulocyte count, and normal or increased iron stores. Soluble transferrin receptor testing helps distinguish from iron deficiency anemia. In the next video, we will discuss EPO resistance, converging mechanisms, and the diagnostic lab pattern.